Welcome.
The Pathology Innovation Collaborative Community is a regulatory science initiative that aims to facilitate innovations in pathology, as well as advance safety and effectiveness evaluation. The PI Collaborative Community aims to harmonize approaches to speed delivery to patients through collaboration in the pre-competitive space. The PI-CC is open to all stakeholders, public or private, including but not limited to academia, industry, healthcare providers, patients, and advocacy groups. Meetings of this kind are an example of lawful activity under federal antitrust law, because the focus of the meeting is on government action or policy, including the industry's responses or positions taken with respect thereto. However, antitrust monitoring is needed for companies' own protection, since they are direct competitors meeting together. These meetings need to stay within protected subject matters and need to be monitored so that they do not stray off into inappropriate areas, such as pricing and price terms, sales and service territories for particular products, customers and customer territories, each company's individual decisions regarding selection of suppliers or customers, marketing plans and especially future marketing plans or new product offerings, other proprietary or competitively sensitive information. Today's meeting will follow the written agenda, and there will be no discussion of pricing or other prohibited topics. If the discussion veers into those topics, we will terminate the discussion. If you have any questions or concerns about the propriety of a discussion, please raise it immediately, and we will take a break to determine how best to proceed. The Medical Device Innovation Consortium acts as the formal convener, and we will follow their conflict of interest policy. For additional information, we defer to our Code of Conduct and our website, www.pathologyinnovationcc.org. We look forward to moving the field forward.
Is this correct? Now it is. Hey Emily, thank you for joining.
Hi.
We can probably get started because there's very few people joining still and want to make sure that we can stay on time. If you don't know where you are, you're in the Circulating Tumor DNA session Out of the Dark with Pathology Innovation Collaborative Community. And I wanted to get started here. As you know, the field is booming. This is an overview, absolutely not complete, of various commercial entities that are related to circulating tumor cells or all kinds of floating particles, including ctDNA, exosomes, etc. I wanted to give a thanks and a quick pointer for those who are not aware of the Friends of Cancer Research, a regulatory science and patient advocacy group in DC that has a lot of projects in this realm. I point out to a few. One is the CT Monitor project for monitoring treatment response. They have also produced a fantastic white paper last year called Assessing the Use of ctDNA as an Early Endpoint. I see Brittany is just joining. And then yes, if you had a chance to join their interesting webinar on expediting drug development with use as ctDNA as an early endpoint, you will probably recognize that some of these themes are well aligned. So a brief outline of what we're going to be talking about today is provided here. I think you may have seen this already. But the main point that we want to consider, which is the leading question here, is there a need for commenting on a recent draft guidance? So brief interest in a market as evolving and as complicated as circulating tumor DNA, of course, raised awareness with the FDA. The regulator has established several modalities to solicit input, and one of those is called the Pathology Innovation Collaborative Community, which is a regulatory science initiative. We are providing mostly information to the field. We try to facilitate innovation. Of course, a regulatory science initiative, we deal with things like how can we potentially provide data or evidence to advance safety and effectiveness evaluations. And you can read here that we're acting in the pre-competitive space. So antitrust monitoring fully applies. That means specifically you can't talk about your product's pricing, customer territories. But what we can do is to try to harmonize all kinds of things. And the one thing that we're doing today is to look over a draft guidance and consider whether the field, as it by the many stakeholders on the call, want to potentially contribute or comment or have other components. So we are open to all stakeholders. If you haven't signed up for becoming a member at Pathology Innovation Collaborative Community, Ulla will share a link to the website. It's free and you can leave and join at any time. Quickly, how this works. Typically, people from the community propose projects. In this case, the draft guidance was delivered to us and there was a question, can we make a project out of this? So once you propose a project, it will be presented at the steering committee. There's one happening next week, and then we help solicit input. So you're currently at this stage where a network of people interested in a certain topic can contribute. We generally do not change the scientific content, but we just solicit input. So anyone who will want to comment later on, feel free to use the chat. Any of the other communication options, raise your hand, just speak freely. This is open to everyone. So briefly, this project, which is hovering around a draft guidance that Ulla will paste in the chat that I hope you have read, that came out earlier this year, is that we want to go through this, provide a quick review. This is by no means related to any official statement by the FDA. This is just what several people have contributed so far. So we want to inform the community, provide all the material that you will see today free on the website as a resource, and then enable, as just outlined, networking. That doesn't have to be resulting in a final comment. It can be positive or negative feedback to the FDA or to this guidance, or it could be a publication or any other regulatory science output that you can think of. Questions so far? It's all basic and prelude. So when looking at the guidance, and I can show quickly, this is the draft guidance that we're talking about. To those in the field that know the complexity of circulating tumor DNA, it may come as a surprise that when this was released in May 2022, that this is only 11 pages. So it'll soon be clear why it's much more than 11 pages, but this is sort of a summary. So to outline how these guidances are usually produced. So there's a very complex FDA internal process until a draft guidance is released, and then there's usually, depending on what kind of guidance, a time period where commenting is allowed. So currently this is still in the draft guidance stage. So it is distributed for comment purposes only. So this is not legally binding. This is currently basically to be by stakeholders. And this is the current structure, 5 main sections with a couple of subsections. And I want to briefly go over this and provide sort of an outline what the draft guidance currently looks like. If there are any gaps or where you see that there are glaring errors in your perspective, please again chime in or take notes and we can discuss this. So the first part is the introduction, and I think the 3 main points here are the following. The FDA distinguishes between circulating tumor DNA versus any other circulating DNA elements. So cell-free DNA versus circulating tumor DNA is already distinguished in the background or introduction section. The second point here is that the scope includes investigational new drug applications. So IND is basically covered within this draft guidance. And then the third part, which is of course an existing work and guidance, which is that this current draft guidance refers to the guidance on hematologic malignancies, in particular the regulatory considerations for the use of minimal residual disease in, of course, the development of drug and biological products for treatment. So this guidance is basically referred to in the introduction section as that it should be considered when discussing the main guidance for ctDNA. So in contrast, that's already more than 11 pages, and I believe Ulla has pasted this particular guidance. As a general rule of thumb, and I want to just briefly show this to you as sort of a quick regulatory trick if you're not aware of this. Let me just quickly put this over here. So if any of these guidance documents are not known to you and you're looking for it, so for example this hematologic malignancies, this link, and these guidances relatively quickly using this particular FDA guidance information tracker. So that's a good resource to have, and for all of these guidances and draft guidances, that link should serve you well to find the primary resource. So some of the documents that were shared with us have highlights, etc. If you want to use the primary, that's the best resource to do. The second section provides some overview over the background of ctDNA. First and foremost, the FDA recognizes that ctDNA as a biomarker, they don't specify what kind of biomarker, but ctDNA as a biomarker has regulatory potential. What that means is, and they list one in particular, that ctDNA as a biomarker may assist and potentially expedite drug development. So that is potentially the main use case from a regulatory perspective. Interestingly, and I highlighted this, and we get to this in 1 second, they emphasize the use on early stage cancer settings for various reasons, and that's probably something that we should discuss in this group. They mention enrichment strategies for trial, meaning pre-identification of patients or substratification strategies. They specifically highlight that the detection of minimal residual disease in several studies confers poor prognosis, and they cite at least 6 different peer-reviewed publications. And they consider, at least from a conceptual point of view, residual ctDNA, for example, after resection, as so-called molecular residual disease. I find that very interesting that term is used. In several publications, this is proposed, and there's definitely some reviews that cover something similar. But I wanted to point this out, that's in this draft guidance already. ctDNA assessment and technologies. I know several people on this call have even done some studies on this. And of course, the FDA emphasizes the need for further standardization. Now, I wanted to briefly highlight this early stage comment, and that is specifically coming from the draft guidance in this section, where it states that in the early stage cancer settings, ctDNA may be used to detect a certain targetable alteration. Now, we can discuss maybe later whether that's meaningful or not. To my knowledge, early stage targetable is mostly used in EGFR for osimertinib. In the earlier stage cancers, all the rest is mostly investigational, but that fits with the overarching scheme that this is for investigational new drug applications. So again, just one of the points, maybe this is to some of you also too restrictive, but it is definitely, emphasized here. Is entitled The Development of ctDNA as a Biomarker for Regulatory Use. And it comes in 4 sections or subsections. One, as ctDNA for the patient selection based on molecular alterations, which is implied, and we get to this in a second. This second subsection here is molecular residual disease for patient enrichment, and then 2 things that we get to in a moment. So importantly, this third main section of the draft guide refers to 2 additional guidances. One is the final version of the clinical trial endpoints, but it's important when you look over this. So this was finalized in December 2018, but it is indirectly also using some of the original version of the draft guidance for multiple endpoints in clinical trials. Now I'm pointing this out because the final guidance, clinical trial endpoints, did not account for all the components of the original draft guidance, which the agency thinks about multiple endpoints. And in this section, number 3 of ctDNA as potentially selection or as measures of early response is for now, and we get to that in a subsequent slide, is probably considered a multiple endpoint because ctDNA as a primary endpoint is not included in this section. So for now, at least, most clinical trial design has to very likely account multiple endpoints. So this additional draft guidance, although there is a final guidance, is probably of interest. So I definitely recommend sort of linking to the 2, and I see Ulla has pasted this already. Quickly, ctDNA as a measure of response. So this is very important because it's the analyte, it is what's being measured, and looking at these specific wordings from the agency gives almost like a roadmap of what considered the intended use and how that should be worked out. So one element here is that it could aid in signal finding of drug activity. So this is probably the relevant targeted subset, if you want to call it this, or, but this can be interpreted in many ways. So I just wanted to point out that that is written with a certain intent behind it. The second part under the measure of response is that the FDA encourages sponsors to develop evidence regarding the use of ctDNA responses, and here comes the part with multiple endpoints or multiple things in terms of outcome measures, is in addition to or supporting pathologic complete response information after neoadjuvant therapy. So that's definitely already very specific in contrast to the first statement, but these 2 components are again under the section, the development of ctDNA as a biomarker. Now the last subsection in this particular part is DNA as an early endpoint in clinical trials, which is probably for many of the trial sponsors on the call and in the field of highest interest. Now the early endpoint section is pretty long. There's a couple of highlights here, and I want to point out 2 or 3 things. So clearly the first point immediately says that there's more data required to support ctDNA as an endpoint, and it is likely to predict longer-term outcomes. So, disease survival, progression-free survival, or even overall survival. And as part of this, of course, if it's ctDNA clearance, this becomes a quantitative problem. And the FDA points to various statistical criteria that have been proposed for validating such endpoints. Now this pointer, this reference 6 here, is actually pointing to another draft guidance, which is the meta-analysis of randomized controlled clinical trials, etc. You can read this, but as you can tell, the theme is that the FDA is referring across various lines of guidances and draft guidances. And this one here is, in my opinion, helpful to understand what trial design would look like. And I put here that the trial design, the common things like inclusion criteria, exclusion criteria, the methods, the disease setting, etc., has to be there. But it is important, and hence I switched this to yellow, that the FDA is already considering justifying the suitability of pooling studies. So this is particularly interesting for companies that have multi-compounds or multi-marker elements, where you could potentially see that ctDNA works across, let's say, biomarker subsets or sub-patient groups where you could pool your data. They specify also that the patient population needs to be representative of the population, meaning the intended target group, so who and how. And then these red sections here are highlighted because they're somewhat vague, but an adequate number of randomized trials. So that's, of course, largely subject to interpretation what that means. Or, you know, the FDA requests that the analysis should be based on individual patient level data that should allow an assessment of individual level association. That's, of course, you know, a very, very tough thing to do. And you see the other points here, the timing, endpoints, and the effects of missing data should be accounted for. Many of these components are accounted for in the analysis and the various statistical criteria used in other guidance documents. The fourth section is probably most interesting for those with laboratories. So from a laboratory perspective, the FDA considered assays or assay consideration in three components. One, they defined ctDNA assay types. They consider sampling considerations and how to do or what to entail in an analytical validation. Definitely interesting from a molecular genetic or molecular diagnostics perspective. As a quick overview, the tumor types are separated into two different ctDNA assay types. The first one is where you do tumor sequencing first and then select variants to do a tumor-informed assay, meaning there is some sort of lag time in determining the tumor sequence-based signature and then customizing or prioritizing certain components to it. The other test that the FDA refers to naive is where you have an assay. It can be a fixed panel or a flexible panel, but it is tumor agnostic. So you would run the ctDNA assay without any knowledge about the underlying tumor's sequence. So I think that's a very good sort of regulatory paradigm that's here portrayed. And they already mention in immediate limitations. So they say that the sensitivity specificity in the tumor-informed type of ctDNA assay depend on clinical cutoffs and analytical sensitivity. And they even go as far in the tumor-naive setting to mention whole genome sequencing could be potentially used in a tumor-naive fashion, but that there are certain other components. They even go to epigenetic alterations, methylation, etc. And I mentioned here in sort of a side sentence underneath this section that multiple markers on a candidate gene panel could help assure that minimal residual disease assay will serve its function. Now, whether the last part, even with the development of additional cytogenetic changes, is true or not, I don't know. That's maybe going a little far, but we can discuss. The second part about the assay, or about the assay considerations, pertains to sampling. Now, I believe this is a very, very important point for trial design, and they are very specific regarding several things. So first of all, they mention that shedding is affected by histology grade and stage, and of course the size of the tumor. The timing of the ctDNA testing should therefore be accounted and made specific to a setting. So for two-stick approaches, that will be very tricky and will probably take some time to develop, but I just wanted to point out that was sort of one of the elements in the sampling section. The setting of time points is a very tricky one. As you can tell, there is specified that multiple time points should entail a scientific rationale and potentially provide predetermined and or discussions with the FDA. So if you have a scientific rationale for multiple time points addressed and should probably be accounted for. At the same time, they specify that these time points should be same across study arms. Now that is, there may be trial designs where that is intentionally not the case, but as written right now, the draft guidance says that the time points for ctDNA assessment should be same across study arms and later on also across study sites, and that there is a baseline pretreatment sample should be collected. That's very strong because that may not be in all kinds of trials and or designs. Now, probably the most tricky section in this document is the analytical validity or validation section. And you can tell this is also a section that goes over multiple pages. And interestingly, aside from the statement that is highlighted here, where it basically states that the typical measurements or key performance metrics, sensitivity, specificity, accuracy, precision, etc., should follow existing technicals and standards, they refer to, in this case here, with reference 7, to a safety and effectiveness data sheet, in this case from Garden 360. We can also provide this in the chat, just so people who are not familiar with it know what that entailed. The FDA specifies that the validation approach should entail the entire assay system. So I put here from tube to a market-ready assay that's taken here from the final market assay. How that is possible, what the SOPs are, is not specified, but you can tell they consider an end-to-end sort of coverage, so to speak. The validation approach can differ from tumor-naive versus tumor-informed types of assays that should be implied from what we just discussed in the slide before. And they mention that the validation can happen with clinical samples plus minus contrived samples, depending on the availability. They mention specifically that fixed panels, meaning panels with the content locked down, can be done using cell lines or spiked samples versus personalized panels. It's very prescriptive, but they say that cell lines representing the number and types of variants should be developed. Now that's a great demand. Whether that's always possible remains to be determined. And they also specify that the precision should be demonstrated and reference materials should be developed. Now this is probably also a call to industry to say that the reference material market, especially for regulatory grade reference materials, is improving. And we didn't put here that there's an MDDT pathway for medical device development tools, but one could definitely see some synergies, but that's not included here. Now the last section is the fifth section called Investigational Device Considerations. For those who are running trials, this is probably the most important component, because as long as it's investigational, you can get certain exemptions. And immediately as that this entire guidance is subject to IDE, and IDE is of course under Title 21, and all these rules will apply. So it's clear that if you would go through an IDE consideration, you would have to apply the scope, applicability, etc. of other IDE devices. As part of that, the distinction between significant risk, non-significant risk, or exempt is made. And that by itself goes back to guidance that's referred here. So you can probably read in this how that's specifically defined. And for those who have done this, they know also that the sponsor needs to submit a study risk determination under this guidance. And the study risk determination itself can be submitted through a new process. And there are two. One is a Q submission, which is this guidance document, so that you can assess whether your study risk assessment is okay, so you will get some feedback. Or there is a so-called optional streamlined submission process, which is provided in this additional guidance for industry. So, in other words, the 11 pages of ctDNA guidance starts by referring to minimal residual disease and continues throughout multiple endpoints, as well as statistical and meta-analysis methods, including some existing approvals, and finally to IDE and numerous other submission programs. So this is the quick and dirty overview, this 11 pages that refer and cross-reference numerous other guidance documents. And I wanted to open this up for a discussion, comments, suggestions, and just thoughts now. I know we have a lot of circulating tumor DNA experts on the call. It's hard to speak first, I know. Any comments so far? No comments? What do folk think? Come on. Who should I call on now? I see Tom, I see Jeff, Eric. It's a very shy group today. What's going on? I see a question from Anna. How long will this guidance stay as a draft? That is a very, very good question. That is very hard to answer because some draft guidances are never moved into final stage. But generally speaking, I think it's 90 days. Um, I have to check what the official guidance release date would be, but it's not like a, you know, final date that you can comment. So in case we would want to comment, we could probably go a bit beyond that final state. That answers your question. So the official release date was May 2nd. So 90 days, we're almost there, I guess.
I'll, I'll ask a general question, Joe.
Sure.
Not being my area at all, but it's very interesting. But if you wanted to LDT this, how does this draft document help you?
I think in terms of probably naming conventions, right? That you know what the regulatory terms are. I think there were some papers that called an essay naive, which is sort of a weird naming convention, but that's now at least on record. You can consider some of the analytical validation components that the FDA states as highly, for example, when to use, let's say, cell lines, spiked samples for personalized assays, etc., just to know what the FDA currently is thinking as an, let's call it another independent source of, let's call it compliance or quality in contrast to all the various papers. Some publications mention the number of replicates and how often you should do things. I find relatively informative that they condense down the key elements of timing, multiple time points, endpoints. It's almost more like a resource, not so much as if it gives the final outline. So as an LDT, I think currently the claims that you make or the intended use you would have to specify, and so that the guidance document would provide you at least sort of a framework for what to think about. I know from, let's say, practical experience in the tube, there's a lot of technicalities that are not really covered other than by stating that they consider all of that one essay, so from tube to final report. I'm not sure if that's easy to
That's what I was
realize as an
thinking.
LDT.
And that was I was thinking. I mean, as an LDT, I thought there'd be more flexibility as long as you validate from end to end. But with the FDA under this guidance, it seems much more locked down, very similar to digital pathology. So from some ways, if you want to get this into practice, the LDT is an easier framework to do it because there's some flexibility with things like
So the
as long as you validate. Yeah.
keep in mind the intent of this guidance is not for, let's say, clinical use. They wrote this specifically to help develop biomarkers in cancer clinical trials. So there are other uses of circulating or cell-free DNA, but for cancer clinical trials, it's important that they've tied this all down to an investigational new drug application. So now the big promise for this, and I believe that's what Friends of Cancer Research are portraying and discussing, is of course, can we use ctDNA measurements as an earlier response endpoint so that drugs can enter the market earlier without necessarily waiting for overall survival? That trend has happened over the last decade, from overall survival to progression-free to alternative endpoints. And if you consider ctDNA an alternative endpoint under an IND with the appropriate guidance, as just illustrated, that's what the FDA is currently thinking, what sponsors can or should provide, probably in conjunction, drug and ctDNA measurement, to consider an alternative endpoint. So the scope is not so much how to get an assay into the clinic as can this be used to get a drug into the clinic. What's not mentioned in there is whether that would imply that if an investigational new drug is, whether then that ctDNA assay becomes something like a companion diagnostic or an outcome measurement tool. I didn't see that, at least.
That was that's the direction I would like to take it. Can it be a companion diagnostic? Because there's new drugs, but how about just conventional chemotherapies, other things that are there? This is another companion, another assay you could use it for, because using it as a clinical trial, that sort of slows down getting it into practice in my mind. And there might be some use cases with just conventional monitoring that this could be very applicable. And I'd love to see that into practice. But if it's always tied to an experimental drug, that really will slow its adoption.
Sorry, I'm just typing out the questions real quick so we have that when we share this. I see there's a question from Robin Thomas. Robin, are you on the call?
I am.
Do you want to?
Yeah, I'm just wondering, I've been having conversations around digital PCR versus NGS and trying to figure out when is it appropriate in which setting to use dPCR instead of next generation sequencing. And I wondered if this was maybe an opportunity for dPCR to come into its own, and just wondered if anyone had any comment on that or any opinion around it.
So I think the first part in relation to the draft guidance is they did not specify the method at all. So it's not as if this is specific to NGS or the type of, let's call it PCR underlying it. So I thought that that's a pro. It's more or less discussing sort of the context, not so much the method. Now, when it comes to digital droplet or multiplex digital PCR, the question is whether you lock this panel down or whether you use the flexible technology to adopt quickly. So that would be the types that the FDA defines as tumor informed or naive, and then of course on the scope and amount of markers. But that wasn't specified. If you ask personally, I defer to the to the various experts on the call whether one or the other technology is better or worse. I would say the simple answer is it totally depends on the context, I think, meaning the tumor type, the grade, the stage, your specific application.
Thank you. I mean, I think that's my thinking as well. I just wondered if anyone had any strong opinions or any insight from their own experience. Thank you.
Thank you, Roman. So maybe I can just ask it around what do people think about pointing out a few things about this guidance in a potential comment. Nothing, apparently. It's a very quiet group. So, point out to two other events, because I don't want to hold up anyone who has other things to do than sit on a silent Zoom room. There are two upcoming events next week. One is related to ctDNA as a biomarker in cancer treatment, organized by Friends of Cancer Research. So I think this is one. And then the other one is if you like pathology innovation collaborative community and value regulatory science initiatives, there's a steering committee that's open to the public. I think I can paste the links to both of these so that you can register and or join. And I thank you for your attention, and I hope that this was helpful in terms of providing an overview. So I would definitely invite anyone who has additional questions or comments to feel free to submit that through our website or to Ulla or myself personally. And yeah, if there are no other questions, comments, suggestions, then I think we're done. Thank you very much for your time.
Thank you.
Thank you.